Good morning to everyone present. I'm Aditi Agrawal, third year BDS student. I will be presenting a poster on the topic Involvement of the Oral Cavity and Saliva in Severe Acute Respiratory Syndrome Coronavirus 2 Infection. COVID-19 is an infectious disease that is caused by a newly discovered coronavirus identified as Severe Acute Respiratory Syndrome Coronavirus 2. The oral mucosa and saliva are high risk sites for higher viral loads. Overall, the oral cavity represents a robust and underappreciated site for coronavirus infection. The figure on the left shows a diagrammatic representation. It is a modified image indicating the location of the entry points of coronavirus and the anterior view of the oral cavity labeling different areas of the oral mucosa. Diagram A shows blue arrows which indicate the nasal and oral entrance of the virus. The location of the olfactory epithelium and taste buds. Olfactory epithelium is located on the roof of the nasal cavity. Taste buds can be found in the tongue, tonsils, and oropharynx. Diagram B shows the specific location of the stratified squamous keratinized epithelium and stratified squamous non-keratinized epithelium in the oral cavity. The pathogenesis of coronavirus can be explained by a simple mechanism. Coronavirus enters a host's body and invades the host cells via the ACE2 membrane receptor. This binding leads to conformational changes and cleavage of the S protein from the virion and releases the nucleocapsid into the cytoplasm. The S protein is proteolytically cleaved by cellular cathepsin L and the transmembrane protease serine 2 or TMPRSS2. Coronavirus can induce tissue necrosis factor TNF alpha converting enzyme dependent shedding of the ectodomain of ACE2, and that process was coupled with TNF alpha production. TNF alpha is an inflammatory cytokine produced by macrophages during acute inflammation and is responsible for a diverse range of signaling events within cells, leading to cell necrosis or apoptosis. These data suggest that cellular signals triggered by the interaction of coronavirus with ACE2 expression in the alveolar tissues, oropharyngeal mucosal cells, GI tract, kidneys, and endothelial cells, including oral tissues, indicated that those organs with high ACE2 expressing cells should be considered to be potential high-risk sites for coronavirus 2. The diagram on the right shows a representation of clinical implications and various sources of severe acute respiratory syndrome coronavirus 2 in saliva. A suggested mechanism for coronavirus entry into the salivary gland is shown. The viral spike protein binds with the ACE2 receptor on the surface of the cell, followed by its priming with serine protease, TMPRSS2, and subsequent entry into the cells. After replication and packaging, multiple new virus particles are released from the cells in the saliva, as shown. Despite signs of infection, including taste loss, dry mouth, and mucosal lesions such as ulcerations, ananthema, and macules, the involvement of the oral cavity in coronavirus disease COVID-19 is poorly understood. To address this, two single-cell RNA sequencing datasets of the human minor salivary glands and gingiva, which included 9 samples and 13,824 cells roughly, identifying 50 cell clusters, was generated and analyzed. Using integrated cell normalization and annotation, 34 unique cell subpopulations between glands and gingiva were classified. In the human body, ACE2 receptor, or the angiotensin converting enzyme receptor, is known to be important in regulating the blood pressure homeostasis, and it regulates the renin angiotensin aldosterone system, where it converts angiotensin 1 to angiotensin 2. The single-cell RNA sequencing studies of ACE2 expression have detected high levels of expression in keratinized epithelial cell surfaces of the oral cavity, such as the dorsum of the tongue and hard palate, rather than the buccal or gingival tissues. Saliva samples were collected to conduct a few experiments. Saliva from the coronavirus infected individuals harbored epithelial cells exhibiting ACE2 and TMPRSS expression and sustained coronavirus infection. Acellular and cellular salivary functions from salivary fractions from asymptomatic individuals were found to transmit coronavirus ex vivo. Matched nasopharyngeal and saliva samples displayed distinct viral shedding dynamics, and salivary viral burden correlated with COVID-19 symptoms, included taste loss. Upon recovery, this asymptomatic cohort exhibited sustained salivary IgG antibodies against severe acute respiratory syndrome coronavirus 2. To rule out contamination from respiratory secretion, saliva was collected directly from the opening of salivary glands and found that COVID-19 nucleic acid, suggesting that salivary glands were infected. 4 out of the 13 cases who were nucleic acid positive were positive in saliva, and 3 out of these 4 were critically ill patients and were in need of ventilator support, suggesting that COVID-19 nucleic acid positive in salivary gland originated saliva as an indicator of severity of COVID-19. In conclusion, these data show that the oral cavity is an important site for severe acute respiratory syndrome coronavirus 2 infection and implicate saliva as a potential route of coronavirus 2 transmission. Salivary glands could propagate coronavirus infection and sustain COVID-19 in other body sites. Single cell sequencing is an important tool because subsequent experiments revealed that coronavirus 2 tropism was broadly identified for the ACE2, TMPRSS2 expressing acini and duct epithelial cells. A cellular source of COVID-19 in saliva might be derived from salivary glands, as there was detection of virus throughout the functional unit of saliva production. Thank you.